Hello students, I will be taking class on chromatography today, and this lecture is only for MBBS students. Please remember. Now this is one of the competency that it is 11.16. Observe use of commonly used equipments or techniques in the biochemistry laboratory including paper chromatography and TLC. So we will be giving only in the form of spotter or OSPE. Even we can ask you to define chromatography. What are the different types of chromatographies? You have to tell us. Then what all things are there in this. You should know the principle of chromatographies. Now how you will define this term? This technique was introduced by Russian botanist Mikhail Svet on a separation of plant pigment chlorophyll. So it is a technique for the separation of similar compounds by a continuous redistribution between two phases, that is stationary phase and the mobile phase. So this is very important that you should be able to, you should know the concept of stationary phase as well as the mobile phase. So now coming to the mobile phase refers to the mixture of the substances dissolved in a liquid or a gas. It is a solvent moving through the column. Then stationary phase is the substance which stays fixed inside the column. You can say stationary means fixed. Other way you can, if you need to remember the things. Stationary phase is the porous solid matrix through which the sample contained in the mobile phase percolates. The interaction between the mobile and stationary phases results in the separation of the compounds from the mixture. Now, the basic principle behind this chromatography is that the basis of all forms of chromatography is the distribution or partition coefficient, which describes the way in which a compound distributes between two immiscible phases. So these phases we are talking about the stationary and the mobile phase. So this technique is based on the differential migration of the individual components of a mixture through a stationary phase under the influence of a mobile phase. If it is less soluble in the mobile phase and if it has more affinity towards the stationary phase, so it will retain, or we can say it will remain on the stationary phase. If it has less retention power and less affinity towards the stationary phase and more solubility in the mobile phase, so we can say it will move in the upward direction. So basically these interactions include the 5 physicochemical principles. So all the chromatography, whole of the chromatography is basically based on the 5 physicochemical principles. These are adsorption, partition, molecular sieving, ion exchange and the affinity. So on the basis of these 5 basic principles, we can even call them the respective chromatographies. So adsorption chromatography, partition chromatography, molecular sieving chromatography, and exchange chromatography, affinity chromatography. So even we can ask you the principle of all these basic mechanisms, 5 basic mechanisms of physicochemical principles that are there in the chromatography. So now coming to the classification. So in many books different types of classifications are given. So first of all, when we divide this chromatography on the basis of mobile phase, as you all know that mobile phase, so depending upon on the basis of mobile phase, it is divided into 2 main types, that is liquid and gas. Mobile phase, because mobile phase can be a liquid or a gas, solid cannot be a mobile phase. And on the basis of stationary phase, again they are divided into solid and liquid. Again gas is divided into solid and liquid, because you know that stationary phase is a solid or liquid supported on a solid. So now This is again a different classification of chromatography. This is based on the chromatographic bed shape. We can divide this on the basis of chromatographic bed shape into planar and the column chromatography. So under the planar, paper chromatography is there and thin layer chromatography is there. In the same way here, example of liquid solid chromatography is column chromatography, and liquid liquid chromatography is paper chromatography. So we can ask you that give an example of liquid liquid chromatography. So it is the paper chromatography. Now the first mechanism, that is adsorption. So adsorption is a surface phenomena in which one molecule is adsorbed on the other. Depending upon the adsorption power, separation happens. For example, an alumina here acts as an adsorbent material, and if the compound has more affinity towards this alumina, it will retain in the alumina, while another which has less affinity will move in the downward direction and will leave out through this. For example, solid liquid chromatography. So example is column chromatography. So the adsorbents such as silica gel, alumina, charcoal powder, all these adsorbents we can use here, and calcium hydroxyapatite are packed into a column in a glass tube. This serves as a stationary phase. The sample mixture in a solvent is loaded on this column. So this is acting as a mobile phase here. The individual component get differentially adsorbed onto the adsorbent. The elution is carried out by a buffer system. As I told you, this is a mobile phase. The individual compounds come out of the column at different rates, which may be separately collected and identified. Now the second mechanism is partition. So partition depends upon the solubility towards the mobile phase if compound is moving. If it is soluble in a mobile phase, it will move out fast or earlier. If it is less soluble, it will retain. An example is liquid liquid chromatography or paper chromatography. HPLC is also an example of partition chromatography. Now third mechanism is gel filtration or size exclusion chromatography, that is based on the molecular size. So size exclusion chromatography separates proteins based on their size. So proteins are applied over the beads containing pores. The beads are made up of agarose or the dextrin. Beads with different pore sizes are available commercially. So larger proteins cannot enter the pores of the beads. They move through the available space in between beads and comes out faster, as they will not enter into the pores, or we can say they are not entrapped into the beads, so they elute out faster. But smaller protein, they enter through the pores of each bead. They spend more time in passing through the pores and come later or elute later. So based on this, it is also known as molecular sieving chromatography. Columns are packed with gel-like material. Gel has beads in it, as I already told you. These beads has pores. The smaller particles are trapped in these beads in the gel due to the molecular sieving effect. It is dependent on shape and size of a particle. Larger particle will elute out fast because they are not entrapped inside the beads. Smaller particles get entrapped inside the beads. It is also known as gel filtration effect. So you can see this in the diagram also, that if that protein solution is added on the top of this column, so what will happen, small proteins, they will entrapped into these beads and they will reach the bottom, or they will take longer time to reach the bottom, but the larger beads, they will come out through these spaces and they reach the bottom faster. Now coming to the next mechanism, that is ion exchange, and on the basis of this mechanism, there is the chromatography is known as ion exchange chromatography. So two types of resins are there in this, anion exchange resins and the cation exchange resins. So column is packed with resins and exchange of material occurs in between these resins and compounds on the basis of charge. So it is dependent on ionization. So cation exchange resins will exchange cation and anion exchange resins will exchange anion. So cation exchange resins will retain the positively charged ions and anion exchange resins will retain negatively charged ions. I'll tell you how. So the stationary phase is made up of charged beads in a column. The beads can be either positively or negatively charged. If positively charged beads are used, the technique is known as anion exchange chromatography, since the beads retain the anionic proteins which can be eluted at a later stage. If negatively charged beads are used, the technique is known as cation exchange chromatography, since the beads retain the cationic properties which can be eluted at a later stage. Now you can see this positively charged proteins bind to the negatively charged beads and negatively charged proteins flow through this. So ion exchange chromatography is based on the exchange of ions between a charged stationary surface and ions of the opposite charge in the mobile phase, and depending on the conditions, solutes are either cations, that is positively charged, or anions. These are known as anion exchangers. So anion exchangers are made up of two parts, as I already told you, an insoluble matrix and chemically bonded charged groups within and on the surface of the matrix. It is classified as cationic and the anionic based on whether it exchanges cation or anions. So cation exchanger, also known as acidic ion exchanger, and anion exchanger, also called basic ion exchanger. Each type of exchanger is also classified as strong or weak depending on the ionizing strength of the functional group. Now if you see this is cation exchanger, stationary phase particle, positively charged, attracted to the negative surface, and this is anion exchange, stationary phase particle, so negatively charged and allied, attached to the positive surface. So this is how ion exchange chromatography takes place. Now the next mechanism is the affinity, and based on this there is one chromatography that is known as affinity chromatography principle. Affinity chromatography is based on the property of specific and non-covalent binding of the proteins to other molecules referred to as ligands. For example, enzymes bind specifically to ligands such as substrates or the cofactors. The technique is based on the high affinity of specific proteins for specific chemical groups. Thus, coenzymes can be used to purify the enzymes. For example, NAD is used to purify dehydrogenases. By using antibodies, antigens could be easily separated. So conversely, we can say antibodies can be purified by passing through a column containing antigen. So this is how it takes place. Then coming to the applications of chromatography, HPLC is a gold standard method or analytical process used for the measurement of different analytes. So paper and TLC, that is thin layer chromatography, is used for the separation of amino acids and the reducing sugars, and this technique of chromatography is used for the purification of carbohydrates, proteins, lipids, enzymes, vitamins and the drugs. And this technique is also used for the measurement of concentration of hormones, proteins, enzymes, vitamins, amines and the drugs, etc. And it is also used for the screening of inborn errors of the metabolism. Affinity chromatography is useful for the purification of enzymes, vitamins, nucleic acids, drugs, hormone receptors, antibodies, etc. These two techniques, that is adsorption as well as affinity, it is used for the separation of larger molecules such as enzymes, proteins, glycoproteins, polysaccharides and the nucleic acids, etc. Now coming to the paper chromatography, as this is in detail. So in this solvent system, butanol is moving by the capillary action through this filter paper, and the substance which has more solubility towards the solvent system, it will move upward, and which has less solubility, it will move less as compared to this first one. So by calculating the RF value, I will tell you what is this RF value. By calculating the RF value, you can identify the specific spots. Now in this, the solvent system or solvent system is basically known as, or you can memorize this by the name BAD, that is butanol, acetic acid and distilled water in the ratio 12:3:5. We can even ask you in the OSPI that what is the proportion of this solvent system which is acting as a mobile phase in this. So water is adsorbed on the filter paper, acts as a stationary phase. This is an example of liquid-liquid chromatography, as both mobile and stationary phase, they are liquid. Water is adsorbed on the paper or cellulose by weak interaction. So you should know this also, that how it is adsorbed. So water is adsorbed on the paper or cellulose by weak interactions, mainly by the hydrogen bonds or weak van der Waals forces. Now coming to the types of paper chromatography, as I told you, this is used for the amino acid separation, and this can be ascending type, descending type, radial or two-dimensional. So basically these two types you should know, that ascending type, and you don't have to go in much details because this will not be asked in your theory, but this is a practical parts, so only few auspices can be formed from this. So we can ask you that what all kinds of paper chromatographies are there, or in the viva also we can ask you. So at least you should know that paper chromatography is of ascending and the descending type. Now what are the reagents and chemicals that are used in this? So I am explaining the procedure with the help of this ascending chromatography. So in this, solvent mixture is N-butanol, acetic acid and distilled water in the proportion of 12:3:5. 0.2% anhydrine solution in acetone is freshly prepared, and also we need the Whatman filter paper. Now the procedure: saturate the chamber with the solvent, that is butanol, acetic acid and distilled water in the ratio 12:3:5. Keep the chamber packed overnight. Whatman filter paper is cut into 35 into 15 centimeter sheet. A pencil line drawn about 3 centimeter from the end, lower edge we can say. Application points are marked on the line at equal distances. So on the middle point, a mixture of known amino acids is applied with the help of a capillary. So in this you can see here, this is a chromatographic chamber, this is the solvent. This we are taking ascending paper chromatography. We have hanged this filter paper with the help of a clamp. This is a chromatographic chamber. This is the solvent front, or the that I was telling you, bad butanol, acetic acid and distilled water in the proportion of 12:3:5. So these are the application points. So make sure that these application points should not dip into this solvent mixture. They should be at least 1 centimeter above the solvent mixture, and these application points should be at least 1 centimeter above this solvent mixture. This is the mixture of amino acids. These are the known amino acids. Any amino acid we can take, and on the basis of RF value, by calculating the RF value, we can identify these spots. So on other points, individual amino acids are applied. So 2 to 5 microliter sample with the help of micropipette. This we have to apply this at least 2 to 5 microliter of the sample. Sheet is dried with the help of a dryer. So in the chromatographic tank, put about 100 to 150 ml of the solvent mixture and secure the lid tightly using a seal and let it saturate overnight. Now place the paper strip in such a way that pencil line is lowermost and about 1 centimeter above the solvent, or the paper should stand absolutely vertical. These are the precautions you should take while hanging the filter paper. Replace the lid. Leave it till the solvent ascends almost to the upper margin of the strip. Take out the paper and let it dry. Spray 0.5% of ninhydrin solution. This will give the purple colored spot because of the ninhydrin reaction. So because of this reaction, you get the purple colored spots. So the paper is first allowed to dry in air and then in a hot air oven at 105 degrees Celsius for 3 minutes. Purple colored amino acid spots are seen. RF value of each amino acid is calculated and compared with the standard. So quantification of amino acids may be done by cutting each spot, eluting and performing the quantitative analysis. So measuring color density using reflectance photometer. So what is RF value? It is the relative flow or ratio of fronts. It is calculated by the distance travelled by the solute to the distance travelled by the solvent system. So you should know this, what is the RF value, and on the basis of this value we can recognize a particular amino acid. So applications: simple and reliable detection of amino acid ureas in a single urine. Then single urine test used commonly for the detection of diagnosis of phenylketonuria, alkaptonuria, and the maple syrup urine disease. Later in the protein metabolism, you will be studying this phenylketonuria. You should know it occurs due to the defects in the enzyme phenylalanine hydroxylase, so that the conversion of phenylalanine to tyrosine does not occurs, and as a result phenylalanine will accumulate and is converted to various keto acids like phenyl pyruvate, phenyl acetate, etc., which appear in the blood and the urine, and we can identify this. Then alkaptonuria, this is a rare autosomal recessive disorder of the tyrosine metabolism due to the deficiency of the enzyme homogentisic acid oxidase in liver and the kidney. So as a result, this homogentisic acid accumulation occurs and is excreted in urine. So what will happen? Urine turns dark black on standing. So by performing this, we can find out. Then is the maple syrup urine disease. So in this, what happens? It is also an autosomal recessive metabolic defect, and it is caused by the deficiency of 3 branch chain keto acid decarboxylase, resulting in the accumulation of these in the serum, CSF, and the urine. So you will be reading this in detail in the protein metabolism. So thin layer chromatography, this is an example of liquid chromatography technique used to separate the non-volatile mixtures. Principle is adsorption chromatography. You should know that TLC is based on this principle of adsorption chromatography. Components: TLC plate, that is acting as a stationary phase. These are stable and chemically inert plates. We can even use the glass, plastic, or aluminum foil for this. With a thin layer of adsorbent material. That we can use can be silica gel, alumina, or cellulose that is applied on them. Mobile phase is a solvent mixture. TLC chamber which we need, and it is similar to the paper chromatography chamber. So stationary phase is applied onto the plate uniformly and allowed to dry and stabilize. Nowadays ready-made plates are preferred. With a pencil, a thin mark is made at the bottom of the plate to apply the sample spots. Plate is placed in the chamber such that the sample spots are well above the level of the mobile phase. So this is the same procedure as we have applied for the paper chromatography. Rest of the procedure is same as the paper chromatography. You can see here, this is the chamber, TLC chamber, this is the TLC plate. We have these are the application points. We can say this should be at least 1 centimeter above this mobile phase or the solvent mixture. So RF value of the partition coefficient is the ratio of the distance travelled by the substance to that travelled by the mobile phase in the same time. For example, if solvent has moved 14 centimeter and compound has moved 9.8 centimeter, this is one of the example that how you calculate this RF value. So RF value of the compound is 0.7. So under a given set of conditions, the RF value of any substance is found to be reproducible and is the characteristic of the substance. So advantages of TLC: it is fast. So when we compare TLC and paper chromatography, so this is fast. It has better resolution. Choice: we have a choice between the stationary phases. So applications of TLC: compound identification, for example alcohols, alkaloids, antibiotics, acids, proteins, amines, etc. Separating multi-component pharmaceutical formulations, detection of biochemical metabolites in the body fluids, and it is also used in the food and the cosmetic industry. So I think this much will be sufficient for you to appear in your university exam. Thank you.